Good evening, everyone. We will be waiting for around 10 minutes so that everybody can join in. In the meantime, uh, we will see what we can do. So, we'll be discussing some problems associated with week 10 of the NPTEL course, Genome Editing and Engineering. So, let's wait for a couple of minutes and see if people join in. It's not long. Think, oh, what? Okay, we'll wait for 4 minutes more and then we'll start. Uh, if anybody joins in. Okay, good evening, everybody. Welcome to the 10th session of the live problem solving sessions that we have for the course of NPTEL Genome Editing and Engineering. So this course is taken by Professor Utpal Bora from IIT Guwahati. I am Sanket Mukherjee, a Prime Minister's Research Fellow from the School of Biological Sciences, IIT Delhi. So let's get to the questions directly. So the first question, the process of spreading to other organs is called dash and is a major cause of death from cancer. Is it A, mutation? Is it B, metastasizing? Is it C, tumorigenesis? Or is it D, neoplasm? I'll give you a minute to think about this and then we will discuss the answer. Okay, so the answer to this question is the process of spreading to other organs is called metastasis, and hence it will be option B, metastasizing, and is a major cause of death from cancer. So let's look at the cancer progression. So most cancers, if you know, in human body or in humans, that happens in the epithelial tissues, right? That's why called carcinomas. So usually wherever the cancer is forming, it could be a breast cancer, liver cancer, or any other kind of cancers. What usually happens, that the progression of this disease is quite similar. So usually in a normal body, there would be normal cells of different, cell types, and they will form a tissue and then an organ, and they will have their own functions. But over time, due to various reasons, one of these cells might develop mutation. So when this happens, usually our body, it has a very robust system of clearing out these kind of cells. Usually whenever there is a cell that is misbehaving, not performing its function properly, there are ways in our body in which these cells are cleared out. However, again, cancer cells, they also have devised their own mechanisms by which they evade this immune response, or the killing by the host body. So these they specifically have a very important property, that is they can multiply in huge numbers. So there's proliferation. So once they proliferate, and again, this takes some time. So after proliferation, a good proportion of cells in that particular tissue is now cancerous. What later happens is, uh, when there is a huge mass of cell that comes up, because these cells do not have the property of contact inhibition, they can grow uncontrollably. So when these a huge mass of cells, they grow up, what happens? They require nutrients. Now for that, there is another process which we call as angiogenesis. I'm sure you must be knowing about it. So in what happens in angiogenesis, blood vessels are formed, vasculatures are formed around these mass so that they can get nutrients. However, this is not a very controlled process. It does not happen how it usually happens during the development of human body. So again, these vessels are leaky vessels. They are not formed in a very good way. Now, what happens is after the cancer has grown in size, there's a adequate number of cells, and these are epithelial cells, mind you. So what happens is these cancer cells, they kind of de-differentiate, or we could say redifferentiate into a different kind of cells, which we call as the mesenchymal cells. So this process is called the endothelial to mesenchymal transition. It is again a hallmark of cancer. So when epithelial cells to mesenchymal cells. So epithelial to mesenchymal transition is the EMT, the full form of EMT. So whenever these cells, they differentiate into mesenchymal cells, these cells, they ooze out of these blood vessels that have been formed through angiogenesis, and they then, you know, enter into the blood circulation. Now the blood circulation it has quite a good pressure, and surviving in this environment is also difficult. But yet again, there are few cancer cells who do survive, and then this process where the cell, it travels from one location, one organ to another organ, is called metastasis, and this happens at the later stage of cancer. What is the problem? Let's say if you have a liver cancer, or let's say one person has a breast cancer. Usually if the cancer is uh, you know it's localized to that particular organ, then it is easier to treat. But at a higher stage, what happens, this cancer, it metastasizes, and there is something called tumor homing, where the cancer, it goes to other specific tissues, let's say the bone marrow, sometimes somewhere else, and they will start developing over there, proliferating over there. So killing these kind of cancer cells, it becomes a huge problem, because chemotherapy cannot be directed to a particular organ just to kill out the cells which are present there. It rather is present throughout the body, and killing that number of cells also damages the good cells which are there in the body. So this is the problem of metastasis, one of the major hallmarks of cancer that happens, and this leads to a lot of serious complications that cancer patients have. So that's all about it. So let's come to the next question. M1 macrophages are able to, one, release pro-inflammatory cytokines such as IL-12, IFN gamma, IL-1, IL-23, and INOS. Two, re-educate the DC and CD4 plus T cells. Three, deactivate CD8 plus T cells, and as a result, promote an immune response against the tumor and prevent tumor progression. You have to choose the correct options. Is it A, 1 and 3, B, 2 and 3, C, 1 and 2, or is it D, all the above? I'll give you a minute to think about it, and then we'll discuss the answer. Okay, so let's see. M1 macrophages, they are able to release pro-inflammatory cytokines such as IL-12, IFN gamma, IL-123, and INOS. That is true. They can re-educate the DC and CD4 plus T cells. That's also true. However, they do not rather deactivate, but rather activate. This would be activate the CD8 plus T cells, and as a result promote an immune response against the tumor and prevent tumor progression. So the correct answer here would be option C. So let's see what happens in case of in a tumor microenvironment, essentially. So T cell exhaustion happens. So it's a hyporesponsive state of T cells in a chronic environment with increased inhibitory receptors, decreased effector cytokines, and impaired cytotoxicity. So in a tumor microenvironment, when the T cells, at times they do not function properly. So most T cells which were functioning earlier in the microenvironment are exhausted. So what happens now is that the cancer, it evades the immune response. Now, native T cells activate and differentiate into effector T cells in secondary lymphoid organs. When effector T cells enter the tumor microenvironment, they are polarized into exhausted T cells with decrease in effector cytokines and increase in inhibitory receptors. Subsequently, exhausted T cells may turn to be defective memory T cells or be deleted physically. So that's the thing that happens. So stimulants present in the tumor microenvironment, such as cytokines, chemokines, and growth factors, determines the polarization of macrophages and their differentiation towards M1 and M2 subtypes. M1 macrophages are able to release pro-inflammatory cytokines such as interleukins 12, interferons gamma, again IL-1, IL-23, and INOS. These pro-inflammatory signals will cause inflammation in those tissues, causing cell death. That's the idea. They can re-educate the DC and CD4 plus CD4 positive T cells. They can re-educate them to recognize cancers, or they can activate the CD8 plus or CD8 positive T cells. As a result, promote an immune response against the tumor and prevent the tumor progression. In contrast, M2 macrophages and tumor-associated macrophages increase angiogenesis and formation of tumor-associated fibroblasts. These cells attenuate the immune response in the tumor microenvironment and increase tumorigenesis or tumor progression. The state of tumor microenvironment strongly affects the patient's prognosis. How the patient will be diagnosed and treated will be determined by the state of the tumor microenvironment. So identifying the uh, mechanisms underlying the tumorigenic characteristics of interaction between immune suppressive cells and tumors can reveal novel therapeutic targets for developing antagonists such as monoclonal antibodies and immunomodulatory drug interventions. We'll talk about some of them. Another approach is to fortify already existing immune response or develop new ones through bypassing their dependence on the robust and intact immune system, which becomes non-functional in tumor microenvironment. One of the ways is production of chimeric antibodies, CAR T therapy, if you have read about it. Immunotherapy of cancer has recently proven its potential in numerous clinical trials. So well, one of the biggest advantages of immunotherapy is that compared to the conventional therapy, the chemotherapy that we have right now for treating cancer, it does not differentiate between a tumor cell and a normal healthy cell. So it's a highly cytotoxic compound that also kills normal cells. So that becomes a huge problem. And whenever we introduce these kind of chemicals which cause damage, which cause death of a lot of cells, that even though it clears cancer to some extent, but the extent of or the I would say the remission, the chances of remission, they still remain. So that's the problem with cancer. You can treat it. It might be better in some time, but later what happens? It comes back. So that kind of issues can potentially be solved through immunotherapy. So that's why it's a promising approach. So let's see to the next question. Immune checkpoints inhibitors, ICIs, are designed to reinstate anti-tumor immune responses by interrupting co-inhibitory signaling pathways and to promote immune-mediated elimination of A. Malignant cells, B. Immune cells, C. Tissue cells, or D. Cytokines. I'll give you a minute to think about it, and then we will discuss the answer. Okay, so immune checkpoint inhibitors are designed to reinstate anti-tumor immune responses by interrupting co-inhibitory signaling pathways and to promote immune-mediated elimination of malignant cells. So these are the cells which are cancerous, which are not good for the body. The rest of the cells are not an issue. So let's see what happens. So if we see the functional properties and dynamic changes of immune cells in tumor microenvironment, let's see what happens. So the T cells in the peripheral blood, they infiltrate into the tumor and undergo a functional state transition, possibly driven by the immunosuppressive microenvironment. So at one place, there is the tumor microenvironment, which is immunosuppressive. It has devised its ways to evade the immune response. On the other hand, since the immune response is aware that there is something that is not going right in the body, it injects its immune cells over that place. So these two things, it happens dynamically. Native CD8 positive T cells or CD4 positive helper T cells, they differentiate into transitional state and finally reach exhausted states. So it happens pretty quickly in the tumor microenvironment because they have immunosuppressing environment, while resting T regulatory cells transit into suppressive states in tumor. Such state transitions result in a reduction of effector T cells, yet an accumulation of exhausted T cells and suppressive regulatory cells, both of which are proven to be proliferating and highly clonally expanded in the tumor microenvironment. Now, myeloid cells in blood are mainly monocytes, including CD14 positive and CD16 positive subsets, while these cells tend to differentiate into macrophages and dendritic cells in tumors. The tumor microenvironment sculpts them to harbor immunosuppressive phenotypes, resulting in an accumulation of suppressive tumor-associated macrophages and classical dendritic cells. But a reduction of CD16 positive monocytes and CD and classical dendritic cells, one which actually is functional. In addition, single cell interrogation facilitates the identification of novel subsets of classical dendritic cells and TAMs, that is tumor associated macrophages in the tumor microenvironment, and reveals that tumor-associated macrophage subtypes tend to co-express M1 and M2 signatures, thus inconsistent with the polarization models. Now, The natural killer cells, they exert their cytotoxic functions with perforin and granzymes when activated by the integrated signals of um, activating and inhibitory receptors. Yet they show reduced cell numbers, impaired cytotoxic function, and an impeded orchestrating effect for immune responses, exemplified by the hampered classical dendritic cell recruitment in the tumor microenvironment. The functional defects of the natural killer cells are possibly driven by the tumor cells through secreting immunosuppressive factors and expressing ligands of inhibitory receptors while decreasing the expression of ligands of activating receptors to hinder the new natural killer cell activation. B cells, they also play a very important role in anti-tumor immunity and immune checkpoint inhibitor treatment as B cells and TLSs, that is tertiary lymphatic structure containing aggregates of immune cell, including T cells, B cells, and FDCs, are found to mediate improved responses to ICIs, the mechanism of which involves activation of TFH and B cells. Now, the activated B cells can differentiate not only into plasma B cells to produce antibodies to clear cancer cells, but also into active T cell mediated immune response by presenting antigens to the CD4 positive helper cells that could promote the activate of CD8 positive cytotoxic T cells. So that's how it works. So let's come to question 4. Cancer vaccines take advantage of tumor-associated antigens, TAAs, or tumor-specific antigens, TSAs, to stimulate the immune system, especially a robust and long-lasting immune response of CD8 positive T cells to inhibit the dash of tumor cells. Is it A, growth, infection, recurrence, B, killing metastasis and recurrence, or growth, metastasis, and immunosuppression? Or is it D, growth, metastasis, and recurrence? So I'll give you a minute to think about it and then we'll discuss the answer. Okay, so cancer vaccines, they take advantage of tumor-associated antigens or tumor-specific antigens to stimulate the immune system, especially a robust and long-lasting immune response of CD8 positive cytotoxic T cells to inhibit the growth, metastasis, and recurrence of tumor cells. Let's see what happens with cancer vaccines. So, as already stated, cancer vaccine, it takes advantage of the tumor-associated antigens or tumor-specific antigens to stimulate the immune system, especially a robust and long-lasting immune response of CD8 positive T cells to inhibit the growth, metastasis, and recurrence of tumor cells. According to the clinical use of cancer vaccines, they are divided into two categories. Either they are preventive or therapeutic. So it makes sense. So the preventive cancer vaccines are given in a hope that the cancer will not happen. And the therapeutic ones are given after the cancer is diagnosed. So there are many cancer vaccines now for HPV, the sometimes HCV vaccines. They also work for hepatocellular carcinoma. So there are a couple of vaccines which are already there. So and it's increasing in number because this is one of the ways to stimulate the immune system to recognize cancer and to kill the cancerous cells. So the preventive cancer vaccine, it aims to prevent tumor occurrence by inducing immune response, while the therapeutic cancer vaccines are designed to eradicate the tumor cells by inducing or enhancing the tumor specific immunoreactions. Let's see. The efficacy of cancer vaccine is primarily dependent on immunogenicity, host immunosuppression, preferential expression of tumor antigen, and the delivery of cancer vaccine. The tumor-specific antigens, also known as neoantigens, are ideal cancer vaccine targets due to the highly immunogenic, lower risk of self-tolerance, less common tumor antigen deletions, and lower risk of autoimmune reactions. So far, in phase 3 randomized trials, there is no other therapeutic cancer vaccine yet shown noteworthy clinical efficacy except the CPU-Leucil-T. So while designing a cancer vaccine, you have to take care of a few things. One is the antigen which you will be using to induce the immune response should elicit sufficient immune response. So let's say if we take an antigen that is not a very good immunogen, it does not elicit a very strong immune response, that becomes a problem, because even if the cancer works, while you will have the oh, sorry, even if the vaccine works, even while you will have the cancer, the immune response will not be sufficient to clear off the cancer. That's the problem. Another thing that we should be kept in mind is that while you design these antigens should be of, it should be, ideally they should be foreign, or they should be something which is distinctly different from what is present in your own body. What does this mean? This means is that while you're giving a cancer vaccine, the cancer vaccine should not cause an autoimmune disorder, while some of the healthy cells are also recognized by the body as the cancer cells. And if that happens, then it will cause clearance of those cells, and it is an issue that we would not like to have, uh, you know, um, have in a patient. So these kind of some problems are there, but it's a promising approach. It's a lot of cancer vaccines are being tried, especially the vaccines are given for certain viruses which have links to cancer. So hope they are not cancer vaccines per se, but of course they do reduce the chance of developing a cancer that has been observed already. So yeah, those kind of things are already there. The heavy chain variable region and light chain variable region of antibodies are linked by a small segment of A polypeptide, B CAR T cell, C tumor cells, or is it D CD3 zeta chain? So we'll give you a minute to think about this, then we'll discuss the answer. Okay, so the heavy chain variable region, VH, and the light chain variable region of antibodies are linked by a small segment of polypeptide. So let's see how this looks like. So for developing a chimeric antigen receptor structure, it is composed of a membrane distal single chain variable fragment, SCFV, a spacer domain, and uh, intracellular signaling or activator domain, a transmembrane domain and intracellular signaling or activation domain. In contrast to T cell receptors, chimeric antigen receptors recognize the surface antigens in a non-MHC manner. So yeah, MHC is major histocompatibility complex. Now TCR or T cell receptors identifies intracellular or extracellular proteins that are presented as peptides by the MHC molecules, major histocompatibility molecules. This is the difference essentially between a T cell receptor and chimeric antigen receptors. So T cell receptors, T cell receptor usually it recognizes antigen that is presented by uh, the major histocompatibility complex. That's the idea. This is not in that sense. This does not work in that way. Now, CAR T cells are typically produced by transducing T lymphocytes with a transgene encoding a synthetic antigenic receptor. This transgene is integrated into the T cell genome, transcribed and translated into a chimeric antigen receptor protein, and this chimeric antigen receptor protein is now then expressed on the surface of the T cells. Now, the binding and signaling domains separated into extracellular and intracellular compartments respectively. The extracellular single chain fragment domain is derived from the variable regions of an antibody that recognizes specific tumor antigens together with a spacer that provides flexibility in binding. So that's the idea. The transmembrane domain, it connects the binding domain with intracellular signaling moieties. The TCR-derived CD3 zeta chain drives T cell activation and is fused in tandem with co-stimulatory endodomains that allow for robust and sustained functions. So the extracellular single chain variable fragments work for antibody-like antigen recognition and the intracellular signaling domains for activating the T cells. In CAR T cells, the extracellular domain is responsible for redirecting the specificity of the cytotoxic T lymphocytes to the malignant cells and can be designed according to specific antigens such as the CD19 expressed in B cell, acute lymphocyte leukemia, chronic lymphocyte leukemia, and lymphoma. The, the CAR chimeric antigen receptor intracellular signaling domains provide the necessary signals for priming T cell activation. In the CAR T cell mediated immune response, the single chain variable fragment of the chimeric antigen receptors can engage surface antigens of tumor directly via antibody-like binding. Now, the heavy chain variable region VH and the light chain variable region of antibodies are linked by a small segment of polypeptide. The hinge domain is composed of immunoglobulin superfamily members such as CD8, CD28 or IgG, which play a role in signal transduction. The intracellular signal transduction region is mainly composed of the CD3 zeta chain of the T cell receptor. In addition to the intracellular signaling domains, co-stimulatory molecules such as the CD28 or the 41BB or CD137 can improve cell proliferation and survival time in vivo and enhance the anti-tumor activity of CAR T cells. When CAR T cells bind to tumor surface antigen, they proliferate and they kill the tumor cells. That's the idea. So yeah, that's the answer of this question was a segment of polypeptide. It joins the variable regions, the heavy chain and the light chain ones. So that's the answer to the question. Let's go to the next question. The toxic levels of systemic cytokine release and severe immune cell cross activation in some patients result in the following toxicities. 1. Cytokine release syndrome, CRS. 2. Hemophagocytic lymphohistiocytosis. 3. Macrophage activation syndrome. 4. Immune effector cell associated neurotoxicity syndrome, ICANS. Which of the following option is correct? 1 only, 2 and 3 only, 1 and 4 only, or is it all of the above? Let's take a minute to think about this and then we'll discuss. Okay, so toxic levels of systemic cytokine release and severe immune cell cross activation in some patient, it really results in the following. It causes cytokine release syndrome, yes. It also causes hemophagocytic lymphohistiocytosis. It causes macrophage activation syndrome and immune cell effect associated neurotoxicity syndrome. All of the above will be the answer. So let's see what happens in toxicities that are associated with the CAR T therapy. So the toxicities underlying CAR T therapy have been most extensively characterized in patients in the first FDA approved CAR T cell therapy in CD19 directed chimeric antigen receptors. 23 to 46% of these patients displayed severe supraphysiologic cytokine production and massive in vivo T cell expansion. So that means a lot of cytokine is produced. And if you remember, a similar thing also happened in during COVID. A lot of people who were infected with COVID had a cytokine storm that happened, and that was the reason why they were died, not because of the infection alone, but rather the response of the body towards the pathogen was so severe that it caused fatality. So something like that. So what happens? Toxic levels of systemic cytokine release and severe immune cell cross activation in some patients result in the following toxicities. It causes cytokine release syndrome associated with supraphysiologic cytokine production and massive in vivo T cell expansion. Supraphysiologic means usually in physiological condition, how much T cells cytokines are produced. In this condition, a larger number of cytokines are produced. It's far larger than the physiological condition, leading to adverse effects. Hemophagocytic lymphohistiocytosis or macrophage activation syndrome, a severe hyperinflammatory syndrome characterized by again cytokine release syndrome and combinations of elevated serum ferritin and hemophagocytosis. Hemophagocytosis is essentially the hematopoietic cells uh, or the blood cells. They are killed basically. So that is a problem. If blood cells are killed, it will lead to severe anemia and other kinds of problems. So that is what happens. Renal failure, liver enzymes, splenomegaly. Splenomegaly is enlargement of the spleen, pulmonary edema, and or absence of the natural killer cell activity. Immune effector cell associated neurotoxicity syndrome, ICANS, that is elevated cerebrospinal fluid cytokine levels and blood-brain barrier disruption. So what happens? When this effect is there on the brain, when there are lot number of cytokines in the brain, it causes a large number of cytokines will cause an inflammatory response, and inflammatory response is damaging to all kinds of cells, immaterial to whether they are good cells, bad cells. It's damaging. So the neurons are damaged in this process, so that causes neuro toxicity. So there have been some strategies to overcome these challenges. So let's see what they are. To overcome the barriers of limiting wide applications of CAR T cell therapy, multiple strategies have been developed. One of the most feasible and durable approaches is to generate allogenic universal CAR T cells from the healthy donors. So that is one of the ways. So compared with autologous CAR T cells, the off-the-shelf allogenic CAR T cells have many potential advantages, including immediate availability of cryopreserved CAR T cells for patients in urgent need, enough quantity for first infusion or redosing, and possible standardization of CAR T cells producing. So that's the thing. I think what they mean here is they take it from the healthy donors and they give it back when they need it. So there are banks for this. Whereas considering the presence of endogenous HLA and TCR on donors T lymphocyte, the biggest challenge of universal products via the potential risk of alloreactivity, host versus graft response, and graft versus host disease. Genome editing has been widely used to generate off-the-shelf allogenic CAR T cells and remove the alloreactivity and to address other limitations associated with immunotherapy strategies like TCR cell therapy. So then we come to next question on an application of CRISPR, in which the effect of T cell function on cancer immunotherapy in the presence or absence of a gene is investigated, and the results may be used for the development of further immunotherapeutic approaches is known as loss of functional screening, knocking out granulocyte macrophage colony stimulating factor, immunosuppressive tumor environment, or CAR screening. So I'll give you a minute to think on this and then we'll discuss. Yeah, so an application of CRISPR in which the effect of T cell function on cancer immunotherapy in the presence or absence of a gene is investigated, and the results may be used for development of further immunotherapeutic approaches is known as loss of function screening. So let's see how this looks at what is loss of function screening. So loss of function screening is another application of CRISPR in which the effect of T cell function on cancer immunotherapy in presence or absence of a gene is investigated, and the results may be used for development of further immunotherapeutic approaches. By adopting such study by Schifrut et al., it was revealed that the knockout of some T cell regulatory proteins such as the SOX1, TCB2, RASA2, and CBLB improve both proliferation and anti-cancer activity or capability of modified T cells. To elucidate immunomodulatory impacts of combinatorial role of anti-cancer on cytotoxic properties of affected T cells, and to explore the mechanisms of CAR T cell cytotoxicity, a detailed in vitro loss of function screening by means of CRISPR was conducted recently. So what happens is uh, the function of T cells in the absence or presence of certain genes are observed during um, formation of these kind of T cells. So the idea is to see the effect of certain genes on the cancer promoting activity that could be there, and specifically their role in immunosuppression. So if we can knock out these proteins or we can target these proteins or the genes, then in effect, we could increase the efficacy of the therapy, let's say the CAR T therapy. That's the idea. So this is the loss of function screening, one of the ways to screen and to develop immunomodulatory therapeutics. So the first clinical trial testing the safety and feasibility of CRISPR-Cas9 editing T cell receptor T cells in patients with refractory cancer was reported in the year 2019, 20, 21, or 22. So I'll give you a minute to think on this, then we'll discuss the answer. Okay, so the first clinical trial testing the safety and feasibility of CRISPR-Cas9 editing T cell receptor T cells in patients with refractory cancer was reported in the year 2020. So let's see about the clinical trials. What happened? So several anti-CD19 CAR T cell constructs have been investigated, and responses differ extensively among various studies in patients with hematological malignancies, basically lymphomas and stuff like that, especially refractory or relapsed acute lymphotic leukemia. In 2017, the Food and Drug Administration granted regular approval to axicabtagene, as axicabtagene, Ceolucil, or Yescarta as a therapeutic option to treat pediatric young adult B lymphoblastic leukemia uh, and adult diffuse large B cell lymphoma. Yescarta is a CD19 specific CAR T cell mainly exploited for the treatment of adult patients with relapsed or refractory BCL, B cell lymphoma, following two or more lines of systemic treatment. In July 2020, Tecartus was approved for the treatment of adult patients with mantle cell lymphoma. In February 2021, Breyanzi, the fourth CAR-T therapy for adults with relapsed and refractory large B-cell lymphoma, was approved by the US FDA. CAR-T cell therapy is more restricted in solid tumors than in hematological malignancies, as CAR-T cells are circulated into the bloodstream uh, and lymphatic system, and thereby have more interaction with the blood tumor cells. So usually for lymphomas, we use CAR-T, chimeric antigen receptor T cells. The first clinical trial testing the safety and feasibility of CRISPR-Cas9 editing T cell receptor T cells in patients with refractory cancer was reported in the year 2020. So that answers our question. The safety and efficacy of CRISPR-Cas9 edited universal CAR T cells in vivo needs to be further tested in clinical studies. Currently, 8 relevant clinical trials are going on, and those have been given in your slides as well. So with that, we are ending. We are at the end of the session. I'll just go through the questions once again in case any one of you have missed it. So let's see what were the questions, and then we will call it a session today. So the first question was the process of spreading to other organs is called metastasis and is a major cause of death from cancer. We discussed how the progression of cancer works. M1 macrophages are able to are able to release pro-inflammatory cytokines such as IL-12, interferon gamma, IL-1, IL-23, and INOS, re-educate the DC and CD4 positive T cells, activate CD8 positive T cells, and as a result, promote an immune response against the tumor and prevent tumor progression. Question number 3, immune checkpoint inhibitors are designed to reinstate anti-tumor immune responses by interrupting co-inhibitory signaling pathways and to promote immune-mediated elimination of malignant cells. Then coming to the next question, cancer vaccine takes advantage of tumor-associated antigens and tumor-specific antigens to stimulate the immune system, especially a robust and long-lasting immune response of CD8 positive T cells to inhibit the growth, metastasis, and recurrence of tumor cells. The heavy chain variable region and the light chain variable region of antibodies are linked by a small segment of polypeptide. The toxic levels of systemic cytokine release and severe immune cell cross-activation in some patients result in the following toxicities. It results in all of these: cytokine release syndrome, hemophagocytic lymphohistiocytosis, macrophage activation syndrome, immune effector cell-associated neurotoxicity syndrome. In an application of CRISPR in which the effect of T cell function on cancer immunotherapy in the presence or absence of a gene is investigated, and the results may be used for the development of further immunotherapeutic approaches, is known as loss of function screening. And then finally, we have the first clinical trial testing the safety and feasibility of CRISPR-Cas9 editing T cell receptor T cells in patients with refractory cancer was reported in the year 2020. So with that, we have discussed all the questions that we had today. If you have any more questions, you may definitely ask now. You may unmute yourself and ask if you have any questions. Uh, Otherwise, we'll be meeting again next week on Tuesday from 6 to 8 p.m. So yeah, we are open to any questions if you have from week 10 of this course. And if they are none, then you may leave and we will call it a session. Okay. Okay, it appears that there is no question. So yeah, you may also use the ask question, section that is available on the NPTEL portal to have to ask any questions that you might be having relevant to the course. So with that, we would call it a session today, and we'll be meeting again next week. Thank you very much for joining.